about is the ionic basis of how this is happening. So we're really going to get down onto the subcellular level and talk about ions again. Potassium, calcium, sodium, that fun stuff, right? And how they're moving through these channels to actually create this action potential. So in an autorhythmic cell, we have a different mechanism of creating an action potential than we do in a contractile cell. And that should be apparent, right? The autorhythmic cell can spontaneously depolarize, whereas the contractile cell we mentioned before is receiving an impulse from these pacemaker cells, from these conduction fibers. So we're going to start by looking at the ionic basis of the pacemaker cell, and then we'll look at the ionic basis of a contractile cell. Please make sure you're keeping those two types of cells and the way they function separately or distinctly in your mind, okay? So the autorhythmic cells basically start spontaneously depolarizing by closing potassium channels and opening up two other types of channels. They're going to open up sodium and potassium channels. And these we call funny channels. They're very unique. They're only found in the heart. And scientists who first discovered these call these funny channels because not a lot of other cells, in fact, no other cell in the body, neurons, skeletal muscle included, conduct ions in a way that these do in a response to hyperpolarization. And I'll explain a lot what those mean. But basically, the movement of sodium and potassium here happens via funny channels. Okay, and that's what I'll continue to refer to them as. We also have the opening of calcium channels. Now, there are two types of calcium channels. The first type of calcium channel that opens are slow calcium channels. And then we open up fast calcium channels. So there's another distinction here that we really want to make. There are two main types of calcium channels. We first open slow channels, and then we open fast channels, okay? And then we repolarize the cell by opening up potassium channels. And as I speak about the movement of these ions and channels that are opening and closing, I really want you to not think of this as being super complicated, but think about the permeability. Think about the permeability and how that will affect the membrane potential. If I increase the membrane permeability to sodium, I am going to depolarize the cell. I'm allowing more sodium channels to be open, and so the membrane potential will start to approach the equilibrium potential of sodium, which is very positive. That results in a depolarizing event, right? That's going to be the same case if I increase the permeability to calcium, right? That's going to be a depolarizing event. If I increase the membrane permeability to potassium, and opening up a potassium channel is synonymous to increasing the membrane permeability, right? Don't let these terms sort of confuse you. By opening up more potassium channels, I increase the membrane's permeability to potassium. And what kind of event will that lead to? If there's more potassium moving, what is the ECDF of potassium? Where does potassium want to go? Out of the cell. It wants to get out. We talked about that way back in Unit 1, why it wants to go out of the cell. We looked at the CDF and EDF, we came up with the ECDF, and we know that if you open up a potassium door, potassium wants to leave the cell. If you open up a sodium door, sodium has an ECDF into the cell, so does calcium. They're going to want to get into the cell. So increasing the membrane permeability to sodium or calcium depolarizes the cell, whereas increasing the membrane permeability to potassium is going to repolarize the cell. It's going to make it more negative because we're now approaching that very negative equilibrium potential of potassium. Okay? All right, let's draw this out so that it can make a little bit more sense. So we are studying here the ionic basis, or the basis rather for ionic movement in a pacemaker cell. And bear in mind, this is going to differ for the ionic movement in a contractile cell, okay? these cells can spontaneously depolarize. So we're going to compare on a graph here the membrane potential to time. And we're going to start out at rest. We know rest to be -70 here. And then we know threshold to be roughly -55 or so. Okay. Now, the way that the ionic movement happens in a pacemaker cell is that we start off at rest, and then we're going to slightly increase. We'll talk about what that means, right? Those are the funny channels that are opening. And then those are going to stimulate some calcium channels that further increase. So we have a further dip here. And then we rapidly depolarize, and then we rapidly repolarize, okay? So sort of 4 phases of how this pacemaker cell spontaneously fires an action potential. So let's describe what ions are moving at each of these phases. Oops. Each of these phases. Oops. All right, so let's describe what's happening in each of these areas. So the first thing that happens is that we have a cell at rest. Now, what is the state of our sodium, calcium, potassium channels at rest? We know that we have few sodium doors, right, but many potassium doors. So we have an increased permeability to potassium, a decreased permeability to sodium, and then a decreased permeability to calcium as well. Now, the first thing that happens here is that we close potassium channels, right? And that fundamentally changes what's happening in the cell. So we switch that around. We now have an increased permeability to sodium and a decreased permeability to potassium. And that is because we have closed potassium channels and we have opened our so-called funny channels. Okay. Now, in terms of going deeper into a funny channel, it's allowing the movement of both sodium and potassium. But for the purposes of this class, I really want you to understand it as creating a net movement of sodium. So there's more sodium moving through a funny channel than there is potassium. And so the net effect we see here is that we increase sodium's equilibrium, we increase the permeability to sodium. And because we have closed potassium channels, we decrease the permeability to potassium. Okay, and that's responsible for this initial upstroke here, right? Now, moving forward, what we see is that we increase the permeability to calcium. Calcium is really what's responsible for the upstroke along with this, this brief depolarizing period here to get us to threshold. So we increase the permeability to calcium, and then we further decrease the permeability to sodium here. And what do you think creates this decreased permeability to sodium? So we open up calcium channels. But then we close our funny channels, okay? We close our funny channels. And remember I said that the net movement of the funny channels is sodium. It's a depolarizing event. And so because we had them open here, we had an increased permeability to sodium. But when we close them here, as the membrane approaches threshold, we decrease our permeability to sodium by closing those funny channels, okay? All right, now the third step here is very simple, very straightforward. We increase the permeability to calcium really, this is a really strong increase here in this permeability. And that's because we've opened up a different type of calcium channel, right? These are slow calcium channels. And this is where this gets a little confusing. Slow calcium channels create more of an influence on the membrane potential because they stay open for longer, okay? Whereas fast calcium channels, sometimes called T-type channels, are open very transiently. That's why they're called fast channels, and they don't create as much of an influence on the membrane potential, okay? We're running out of time. Okay, we've got 2 minutes. I'll just finish up this one, then we'll pick it up from there. So we increase the membrane permeability to calcium very, very significantly by opening up these slow calcium channels. Okay, and that is responsible for this rapid upstroke in this cell, which is really what fires the action potential, okay? Now the rest of this is that we flip-flop this, so we open up our potassium channels, because we started by closing them. We're going to open them up here, and then we're going to close the calcium channels. And we'll pick this up the next time we meet.